Okay, so we're going to try and continue where we left off with the endomembrane system and look at two additional players that we think could be of relevance, and both are interesting in what they are trying to do, and both of them essentially originate from the fact that these are vesicles that are coming together to make larger and more complex vesicles which have very unique properties or functions, right? So if you remember last time, we looked at a vesicle that is used for in neurons, a synaptic vesicle, and this kind of the idea of a vesicle and how it could do very diverse things is what we are continuing forward as well, right? The organelle that we are going to talk a little bit about is particularly relevant in context of an entire area of research that has developed in the last maybe 20 years, right? And we'll try and connect it to that. And this area has been well recognized now with the Nobel Prize in 2016 as well, right? And I'll tell you what that area per se is. The organelle that we are going to talk about kind of reminds me of this, right? I don't know how many of you have been fans at some point of time of George Lucas's Star Wars. During the pandemic, I have a 9-year-old son, and we watched, among other things, the entire series. And I don't know how many of you recognize what this is. Right?
It's Death Star. Death Star.
Death Star. That's right. Right. So I think anybody who's seen this, knows this thing. The organelle that we are going to talk about today kind of reminds me of the Death Star. Right. It's a very compact structure that is capable of destroying entire planets, and that's pretty much what the lysosome does too, right? And it's a very potent organelle because it's essentially very dangerous. It's it has the capability, as I said, of destroying much bigger complex structures. And in the cell, the lysosome is essentially a sack of hydrolytic enzymes that digest many different macromolecules. The pH inside the lysosome is maintained by a proton pump in them, and that pH is around 5, though the cytosol is 7.2. And now imagine this is like a bag of acid that is floating around, right? And it has a number of acid hydrolases, right? Nucleases, proteases, glycosidases, lipases, phosphatases, sulfatases, phospholipases, everything that can chew everything else up, right? So it's a very dangerous bag that is floating around. And it's a bag of lipids, right? And it's very interesting how the lipid membrane here is protected from everything that's inside. So the lysosomal membrane, expresses proteins such as, these proteins LAMP1 and LAMP2, which are heavily glycosylated. You remember when we talked about the Golgi, we spoke about the fact that the proteins that make it through the Golgi undergo, among other changes, this change in addition of sugar residues, which is called glycosylation. So they have a number of glycosylated proteins on the surface, which kind of protect them from being attacked by these hydrolases, right? So it's a mechanism which ensures that this sac is available, is floating around, and is able to chew on things when required, but is not going around harming itself in the process. as we just mentioned, lysosomal enzymes can hydrolyze a whole bunch of different things. They work around this pH 5, which is why the pH is maintained the way it is. While rupturing one or few lysozymes, has little impact on the cell, things will get diluted and it'll be fine. But a big leakage in the lysosome content can kind of damage a cell very badly, right? They effectively provide a mechanism by which things can get digested, chewed up within the cell, and can be reprocessed right into their primary units, and sent back into the cell, to build new things. Right, so it's a mechanism of self-renewal, of sustenance, that the cells achieve through the lysosome, right? And just to kind of paraphrase where things are as far as the lysosome is concerned, these are membrane synthesized by the rough ER and then transferred through the Golgi. Many of these modifications, as we talked about, the glycosylation changes likely happen in the Golgi, and then the lysosomes, are budded off. And now available to act on different things, right? And they talk to different other vesicular structures that are present in the cell, right? So they, for example, talk to late endosomes. Late endosomes, are arising from early endosomes, which arise from things that are being endocytosed or pinched from the plasma membrane. They talk to or work with phagosomes, which are essentially, again, endocytosed compartments, but which carry, among other things, it could be a way of taking in bacteria, harmful things that need to be, digested. They also work with this very interesting mechanism, which is called the autophagy, right? Mechanism that is operational in cells, where the cells themselves make an autophagosome, which captures, old dead things that are, need to be recycled in the cell, right? An example of that is the mitochondria that is shown here. So depending upon changes that the mitochondria have undergone, it's possible to identify mitochondria to send them into this into the autophagy pathway, and this is a mechanism by which you can break these things down and break them down to primary components like amino acids, and then put them back into the system in the cell, now making them available to synthesize new things. Right, and the autophagy pathway is something that we will talk a little bit about. Right, just to kind of expand on what we spoke there, along with autophagy, we have these food vacuoles, that can come in, along with food particles that need to be processed, which also is something that the lysosome can fuse with. Anything that needs digestion and being broken down, something that is harmful, something that is that needs to be processed as food, can be handled by the lysosome, right? So it's a remarkable machinery that the cell has, and it does really amazing things, right? Now the autophagy pathway, we'll talk a little bit about, because it's rather interesting, and I will share some content that you guys can go read up a little bit about as well, because there are many interesting implications of autophagy, and what began as the identification or analysis of a cellular organelle now has become a pathway or process that is involved in so many diseases, and the fact that autophagy happens or does not happen has serious consequences for cells. So they can fuse with the autophagosome, that essentially is a bilayered vesicle, that a multi-layered vesicle, not just bilayered, which collects components that needs to be digested and broken down. They can, the autophagosome can fuse with the lysosome. It can fuse with things that are brought in from endocytosis and then fuse with the lysosome. Essentially, eventually what happens is the the components of the lysosome enter the autophagosome, and it becomes an autolysosome. So essentially, it has components that need to be broken down. It has the hydrolytic enzymes that are like now available and flushing through this system, and they break everything down, right? And then things are carried through permeases out to the cell, and used by other mechanisms, machineries that are operational in the cell as building blocks for building new things, right? In 2016, the Physiology and Medicine Nobel Prize went to Yoshinori Oshumi, who pioneered the understanding of autophagy. And interestingly, the idea of autophagy, this kind of self-renewal, if you want to call it, is very beautifully captured in this image that was put up by the Nobel Committee when he was awarded this prize in 2016. So it's a kind of self-eating, right? Something that cells can use to renew themselves and get rid of things that they don't need. And autophagy comes from the Greek meaning self-eating. And is an evolutionarily conserved process by which a cytoplasmic cargo is sequestered in double membrane vesicles and delivered to lysosomes for degradation. Autophagy substrates include organelles such as mitochondria, aggregate-prone proteins that cause neurodegeneration, and various pathogens as well, right? So a lot of this began obviously with the discovery of lysosomes, right? So there is no autophagy, till we know about lysosomes. And lysosomes, interestingly, were discovered by Christian Didouvet, Christian, Albert Claude, and George Palde were all awarded Nobel Prize for Physiology and Medicine in 1974, right? It's 2016, almost 30 plus years between the discovery of lysosomes and the Nobel for lysosomes and the Nobel for autophagy. And it's very interesting how our understanding has evolved over these many years, and how something that was identified as a cellular component so many years ago has now come on board as a player in a major cellular pathway that is autophagy, right? So proteology. So Christian Dubé, when he identified, they did this by self-fractionation methods. You break the cell up and you separate components that were developed by Albert Claude, and found proteolytic enzymes were sequestered within a previously unknown membrane structure that he named the lysosome, right? So the identification of these enzymes is what defined these particular structures. Along with this discovery, Christian Didouvet also found that portions of the cytoplasm are sequestered into membranous structures during kidney development, which they were studying, and recognizing that these structures had the capacity to digest parts of intracellular content. He coined the term autophagy in 1963. Right. So it's very interesting. The idea of autophagy had existed a while, and it took a lot of time to discover pathways and processes. And a lot of that was made possible by Yoshinori's work, where they essentially decided to study autophagy using the budding yeast Saccharomyces cerevisiae, right? And as a model system. And the yeast vacuole he found is the functional equivalent of the mammalian lysosome. And he found that autophagy bodies accumulated in the vacuole when the engineered yeast were grown in nutrient-deprived medium. So when the cell is starving, one of the things it does is it kind of gets rid of things that it doesn't need to kind of operate with minimum capacity. And that's what they used to track down, a whole bunch of different players. So they delineated all the pathways, all the proteins that are involved in making this machinery work, and figuring out how cells actually initiate the process and carry out the process as well, right? So there is this, this beautifully captured. It's a schematic from a Scientific American article, which captures what might be happening, that there is nutrient scarcity. absence of growth factors or low oxygen levels, which could all trigger this. There is nucleation, a double layered membrane called the phagophore, which forms. So this is two bilayers, right? So it's that are coming together. Expansion and cargo recognition, the phagophore expands and closes in on itself, probably by adding new sheets of membrane. It then surrounds and engulfs a built up the cytoplasm along with perhaps a damaged protein or an organelle. And so it kind of wraps around this cytosol with proteins or with cellular organelles. And then things are, you have the autophagosome that is created. The double layered membrane seals, and the resulting autophagosome sheds membrane proteins that took part in its formation, and the proteins are cycled back. So some of the proteins that are required for making them are lost, and now this essentially goes and fuses with the lysosome, right? And the lysosome delivers all the proteolytic enzymes which go in and digest, and then the cargo, the broken down cargo, is now recycled back into the cell, right? So so that's the really interesting role that the lysosome particularly has in cells, and it's very interesting that and I haven't discussed this in detail, but a couple of years ago in 2016, when this Nobel Prize was awarded, there is a talk I gave that is on autophagy and the discovery of autophagy itself. It's available on the Science Media Center's website, YouTube site. You can go check it out. And it talks a little more in detail about autophagy, what kind of disease conditions it's involved in. And I'm not going into all those details. Not everybody's going to be interested in that. But if you're interested, please go look this up, right? So the point that we wanted to make about the lysosome here was that there is no autophagy unless there is lysosome. And these organelles, therefore, have had a significant impact on a mechanism or a process that now has wide-ranging implications for physiology and disease per se, right? The other interesting organelle that we wanted to kind of touch upon at the end is our vacuoles. And vacuoles, interestingly, are vesicles. They are like, there are some studies that suggest that, these are essentially vesicles from the ER and Golgi that come together and fuse to make, bigger vesicular structures. There are different kinds of vacuoles, right? And vacuoles, interestingly, can have slightly different functions, right? So their size is an important factor. The fact that they are bigger than most other, vesicles that are moving around inside the cell. So for example, the synaptic vesicle that we saw, these vacuoles are going to be considerably bigger. They could be a way for the cell to pick up food. So there are food vacuoles, that are initiated or that are created by phagocytosis. And these again then fuse with the lysosomes to kind of digest the food per se. There are contractile vacuoles found in freshwater protists, which pump out excess water from the cell. We have structures like this, for example, in paramecium. And on the left is an image of paramecium, and you can see the contractile vacuole, right? And it's indicated in the schematic on the right as well. So these are considerably bigger than other small vesicles. That's the first thing that you notice. They're again made up of membranes, and they have very distinct functions in being able to kind of pump out fluid from the organism itself, right? And then we have the central vacuole, which is essential integral part of the plant cell, and plays a number of very important roles, particularly in the plant cell, right? So the vacuoles, the membrane surrounding the central vacuole, which is the vacuole that is present in plant cells, and I'm going to show you a schematic of what we're talking about here, right? This big central vacuole that is present there. It is made up of a membrane that's called the tonoplast. And it is selective in its transport of solutes into the central vacuole. So it can regulate what comes in, what goes out. in a cell, along with processes like diffusion that actually move things around the cell. There are active mechanisms of transport, of movement that can happen across the membrane as well, right? And vacuoles are a good example of how that can control not only the size of the vacuole, but it can also affect the content of the vacuole, what's stored inside and what's kept out, so to speak. So the function of the central vacuole includes stockpiling of proteins or inorganic ions, depositing metabolic byproducts, storing pigments, storing defensive compounds against herbivores sometimes. It increases the surface to volume ratio of the whole cell. So the vacuole can grow in size and essentially grow the size of the cell, right? And at least in plants, there are two types of plant vacuoles. The protein storage vacuoles of neutral pH and the lytic vacuoles of acidic pH, which are equivalent in function to the lysosomes as well. So it can act as a storage organelle, as a degradative compartment, as an economical way of increasing size, right? So when, for example, water is added, to plants, there is an increase in the amount of content that is present, fluid that is present in the vacuoles, allowing them to grow in size. That also makes a significant impact on the size of the cells. And remarkably, in plants, these same vacuoles are also used as a controller of turgor pressure, which is the osmotic pressure that pushes outward on the cell wall and keeps the plant from wilting, right? So it also ensures that when you add water and the leaves perk up, remember that's turgor pressure, and that turgor pressure is mediated by this very vacuole, right? So it's really interesting how these when you think about the endomembrane system and you think about the fact that they are all membranous bags, right? How differently these bags of membrane have been adapted, right? We saw the ER where the smooth and rough endoplasmic reticulum is present and protein synthesis happens. We saw this very nice arrangement, which is the parking lot arrangement for the ER. Same kind of membrane put together, assembled in a very distinct way, along with ribosomes to do something, right? We had the Golgi, where everything is kind of nice and stacked, and things move from one compartment to the other, and processing takes place. Enzymes are kept apart from each other in the way the Golgi is built, and effectively things are processed, and from the trans-Golgi network, budding of vesicles is taking place. We saw this movie about how clathrin, which is a very beautiful mechanism for endocytosis, allows or supports the budding of vesicles from the Golgi. So, and then things are carried, right? And among the things that are produced from the Golgi are again these bags of lysosomes, right? Which we now know are involved in binding to food vacuoles, in binding to the autophagosome, in binding to the mechanisms that drive autophagy, components that drive autophagy. All this is, and then you have vacuoles, right? Like this plant vacuole that essentially can change so many things about how plants work, including something as fundamental as turgor pressure, right? So same idea, same components, a bag of lipids containing something inside, keeping some things outside, right? Having a certain composition, and the membrane of that organelle affecting what is retained and what is not present in that particular organelle. This is the basis of all these components, right? And they're all talking to each other. They work with each other. Things get delivered to very specific points. These organelles can fuse with each other, all made possible because of the fact that there is this lipid bilayer around them, right? And that's the really remarkable thing about these structures, about the endomembrane system at large, that they come together, they work together in this very elaborate, intricate manner, and in doing so, they are able to drive so many complex pathways that work in the cell. In the next class, what we are going to focus on is something where a lot of this begins for the cell, right? And we are going to focus on the nucleus, which is this big yellow dot at the center of the cell. Where, now that we began from outside, we've come all the way in, right? And we've looked at all these components. We've seen the complexity with which they operate. We've seen the kind of molecular crowding that exists in cells, cytoskeleton, cytosolic proteins, membrane vesicles, Golgi, ER, all this in a very tight, compact structure, and doing their own thing, talking to each other, recognizing what's happening to the other, and then allowing for the cell to function on a minute-to-minute basis, right? And if you stop and think about this, it is really remarkable and staggering that this network is doing this every single minute that you and I are talking to each other, right? And it's what's allowing for everything that we experience on a day-to-day basis to actually be possible, right? So that complexity is what I hope we will also take away from this, that as we break this down into simple pathways and processes, we know what the microtubule looks like, we know what the lysosome looks like. We also step back and think about how all of this works in a very intricate, elaborate architecture that is the cell, right? And that's what we are, at least this set of lectures is aiming to do, is to kind of introduce you to all these players, let you see what each one of them does in their unique way, and also lets you appreciate the complexity that is in operation at any given point of time, right? So we'll pause here with the endomembrane system.